For this activity, you're going to need a microscope, a microscope slide, a cover slip, forceps, a clean swab, a couple of paper towels, and a drop of bottle of methylene blue. First thing we're going to do is lay out the paper towel to work on. This is not totally necessary, but it will help us to see the glass slides better and will also stop us from getting the desk stained if we overdo the methylene blue later on. Your next step is to take your clean swab and rub it against the inside of your cheek. To make sure you pick up plenty of cells, this needs to be done for an uncomfortably long amount of time. Once you have a swab full of cheek cells, then we can spread them in a circle about 1.5 centimeters in diameter on the middle of the slide. Once you have finished with the swab, make sure it goes in the disinfectant beaker on the teacher's desk. Back to the slide, on top of the cells we can add a single drop of methylene blue. This will dye the cells and make their structures easier to see. To finish off our slide, we need to add a cover slip. This is an extremely thin piece of glass, so it needs to be handled with care. Best way to get the cover slip onto the slide is to place one edge down first, hold the other side up with a pair of forceps or a mounted needle. Then you can gently lower the slip onto the slide. There will always be some excess dye under the cover slip. This needs to be removed before we will be able to see our cells. We can remove it by gently touching the edge of the slip with the edge of a paper towel. You should be able to see the excess dye soaking into the paper towel and the slide starting to look clearer. Once no more dye is seeping out, then our slide is done and we can put it on the microscope. We always start with the lowest magnification, which is the shortest lens, and work our way up. If everything has been done correctly, this is the sort of cheek cell images you might see. At the lowest magnification, the cells just look like small blue specks. Increasing the magnification to 100 times starts to reveal some of the structure. Going on to 400 times, you should then be able to see the nucleus of an individual cell.